Okay, good afternoon, everybody. I think well, depends on when you actually view the seminar, but our on Q&A sessions will be in the early afternoon, right around lunchtime. But anyway, my name is Zhiqiang. I'm a professor at UH-CTAHR. So my lab has been working on coffee leaf rust control project for the last 3-4 years, 2-3 years. And today I will be talking about our research update on field trials of systemic and biological control fungicides for the management of coffee leaf rust. This is specifically to our USDA SCRI project. And I also want to acknowledge my co-authors, Elizabeth, Julie, Zhening, and Nina for their contribution to this work. Okay, let's go ahead and get started. Okay, next slide. Okay, so we don't have to spend too much time introducing the coffee leaf rust because everybody who are interested in this webinar series probably are aware of the coffee leaf rust already. First confirmed back in 2020 and then spread across the major islands, and very bad fungal disease for coffee worldwide. And the current control options in terms of fungicides, we are talking about systemic or contact or biological control fungicides are relatively limited. So we do have some copper-based contact fungicides and a translaminar fungicide, and I believe a fungicide as well. But so far we have not have any true systemic fungicides registered in Hawaii. This is where our research actually came in. We want to identify effective systemic fungicides and based on the result, and we want to communicate and basically convince the manufacturers of those fungicides to register their products for Hawaii's coffee grower use. Sorry, my USB drive is having problem. I'm going to redo this. Well, not sure what happened. I'm gonna have to stop sharing first. And okay, well, let me gotta reshare this. Okay, and okay, I hope I don't have to do this anymore, but sorry, yeah. Yeah, if I have to do this one more time, I will just stop and record. Okay, so symptoms and signs, and we don't have to go through this too much again. And I'm sure Andrea's education program has provided a lot of details on a lot of this signs and incidents and the diseases. So we will just jump into our research. And I manage the R4 program at UH-CTAHR. So this is a specialty crop pest management, and many of you guys probably know Julie and James. They have been our long-term, R4 employees, associates for the past 30 roughly years. And for your information, Julie just retired last December, actually still last month. And we are getting a replacement for Julie. That was actually good news. So we are going to have another technician position replacing Julie soon, hopefully after, well, soon into the near future. But right now, since Julie retired, we only have 3 people working on R4 trials. That's myself as a PI, and James and Ning, who is our technician. So then this is our, we from a 4-man team down to 3-man team, but we will be back to 4-man team again, hopefully soon. So for this research, though, it's not directly part of our R4 project, but many of these things are, because I manage both R4 projects and this SCRI project. So then, we did this SCRI CLR field trials in 2 years, basically year 1 from 2022 to 2023, year 2 the next year. And our goal was to repeat in the second year, basically repeating exactly the same treatments in year 1 so that we can provide robust data to support or not support certain chemical use to control coffee leaf rust. That's myself, and this is Lily, who graduated last year, but she did collect majority of the coffee leaf rust SCRI project data for 1.5 years. So I would say she's a key person for a lot of this data collection. So as you can see, we don't have to go through all these treatments in details, but we were testing 3 systemic fungicides and 2 biocontrol fungicides. One is plant extractions. The other one is a bacteria-based product. And we ran this trial repeatedly, basically 2 years. First year, second year, repeating the first year same treatments, both in Corner Hills, one of the major coffee farms in Kona area. So this is the field layout. To make the long story short, we have 3 replications under each treatment, and we did this totally RCBD randomized complete block design. And as you can see here, within each block, this block 1, block 2, block 3, we have all 6 treatments, 5 treatments plus untreated control, and we have 6 coffee trees within each plot. But we were only taking the data from the inner 4 coffee trees within each plot, so that we can minimize the potential, border effect between any 2 adjacent plots. So that was the data collection, and this is basically the detailed application schedule based on the manufacturer's recommendation, and we don't have to go through all of these details. But some of these, treatments, most of them we apply them according to the manufacturer recommendation. It was 3 or up to 5 times overall in total. So data collection in each of these under each treatments, we are collecting 2 main variables. One is the instance of disease, basically how many leaves had a coffee leaf rust infestation on them compared to the total leaves in that particular branch. And we took 4 trees, like I mentioned, for the measurement, and we randomly selected 4 branches per tree, 2 on both sides, and we counted how many leaves are infected with coffee leaf rust fungal, pathogen. And over that divided by the total number of leaves on that branch, that's the disease instance of disease. And also we did a rating system, so severity of the infestation. So that's from 0 to 5, as you can see. 0, 1, that's a definition of the rating, the 5 rating system. And we also document any fatal toxicity effects on these treatments, if any. But we did not observe any fatal toxicity or crop safety potential concerns for both years. So that was good news. So jump into the main conclusion. We have a lot more data, but I just want to show you 4 slides. Those basically summarized all the key points of these 2 years research. Year 1, as you can see, we did an entire year, June 2022 to May 2023. So that is 1 year, right? So then we, as you can see, to try to convey the key message here is most of these fungicides, they were working effectively for several months in the beginning, but then only about, this is the red line here, was able to maintain to keep the coffee leaf rust instance very low, most of the time under 5%. The 5% is arbitrary, but many people recognize that 5% instance, if anything above 5%, it's relatively challenging to manage or control. So then, as you can see, Abound was able to suppress, to keep the instance under 5% for most of the time until towards the very end of the 1 year cycle. So it kind of become the effect faded away. But I know many folks are interested in the 2 biocontrol products, and they did provide some efficacies, protections against the coffee leaf rust, but they were not as effective as the Abound systemic fungicide based on this data collection. And similarly, we are coming back to that rating system 1 to 5, and as you can see that most of the treatments, they were providing some efficacies, providing some control capacity, but still again, Abound was able to maintain all the instance down below 1, the level 1, which is a very good control efficacy. So that's year 1. So this is a quick summary. So we don't really have the time to actually go into all the details, but the key message for year 1 is everything worked for several months, but Abound was effective, most effective, and has the longest treatment longevity compared to any other treatments that we tested in this 1 year trial. And year 2, as I mentioned, it's exactly the same repeat as year 1, because we want to be able to generate 2 years data. So to showcase that, okay, so 1 year we have this data, and in year 2, if we have similar data and then we can make very robust conclusion or recommendation for coffee farmers that, okay, so this is indeed working in both years, and it's likely that it will work, in the next years, right? So that's kind of like the idea. And many, research publications that require actually 2 years data anyway. So then that's what we proposed, and we did that, just that. And as you can see here again, Abound was again the best treatment among all the treatments we tested. And again, those two biocontrol fungicides, biopesticides, they are effective for several months, but then their treatment effect faded away much quicker than Abound, which is the best treatment identified in this case. This is the percent instance, the disease instance. And the second variable is average severity rating, the rating system from 0 to 5 scale, right? And again, as you can see, Abound was the best treatment, was able to maintain, to keep the rating, the severity rating scale below 1 for most of the time. But then later on, after 7, 8 months or 9 months, the treatment effect faded away, which is more or less expected. So then several things I want to mention. As you can see, towards the very end, everything dropped. So that is not to say that all the treatments are becoming suddenly everything is now effective. Not, towards the very end of the study, which does not make any sense. So the reality is that towards the end of the study, many leaves actually dropped from those coffee trees that we used for the research, and that kind of gave us this superficial reduction of both the severity rating and also the percentage of disease instance, which is actually not necessarily reflecting the reality that the treatment efficacy was extremely effective towards the very end of the treatment 1 year cycle for both years. So that's one thing I want to make it clear. It's not all of a sudden magically every treatment is extremely effective towards the end of the study, end of the 1 year. No, it was because of most of the leaves got dropped. Second point I want to make is probably more important is that, as you can see, both years, and we have identified that Abound was very effective. Almost throughout the 1 year cycle. So that is a very important finding, based on this 2-year research. So key summary, although we did not include Excelia, which is another true systemic fungicide, but we did include Excelia in 2 years of our R4 trial, which I did not provide any graphs or any summary of results, but I do want to mention it in the overall summary that Excelia and Abound both were very effective against the coffee leaf rust in our multiple years field trials. Excelia now is the manufacturer Valent is pursuing the Section 3, which is the federal level fungicide label for coffee leaf rust, so which is in the process right now. Abound is manufactured by Syngenta. So if anything, Syngenta will be willing to pursue the special local need label for the coffee leaf rust control in Hawaii. So these two are both making progress in terms of be able to fully or officially, legally used by coffee farmers here in Hawaii. And again, as I mentioned, biocontrol fungicides are always preferred, or people have a lot of interest in it because organic farmers can use it. However, based on this biocontrol, the trials, 2 years, our 2 years data, both years showed that this biocontrol fungicides, they did work. They did provide, several months of protection. However, they are not as effective as the best systemic fungicide that we identified. But that is not to say they are not useful. They can still be used on top of your, rotation, chemical synthetic fungicide rotation programs or whatever you are trying to use. But then on top of that, if you can somehow make use of this fungicide, biological control fungicide, you can, potentially reduce the use of synthetic fungicides. That's how we view this. Alone, they are not going to cut it with, only using them as the biocontrol approach to control the coffee leaf rust. And we believe that this research provided important options for CLR management in Hawaii and other regions where coffee is grown. So what else do we don't want in Hawaii? And we went to R4 National Conference back in, now it's almost 2 years ago, and we observed Puerto Rico. It was the conference was in Puerto Rico. So Puerto Rico has everything in terms of plants we grow pretty much. They grow coffee a lot. and also everything in terms of pests and the fungal pathogens and everything. So over there, they have this coffee leaf miner, which is the insect pest, causing a lot of damage to the leaves of coffee plants. And also they have a coffee berry anthracnose. We don't have it here. We have anthracnose on twigs, but on leaves, but we don't have the berry anthracnose. This is much severe or much worse compared to the anthracnose that we have here in Hawaii on attacking the leaves of coffee. So these two, one insect pest and one fungal pathogen, we definitely don't want them in Hawaii. But we tried, we should stay on top of this, in terms of a quarantine. Hopefully, we can prevent them from coming into Hawaii's, coffee growing regions. I want to acknowledge all my current and former lab members who contributed to some extent to this research, and we work as a team. So sometimes a student, his or her research project is not coffee leaf rust, but sometimes we need extra hands. They need to work on this and that. They help out. That's kind of like the teamwork strategy we use for most of the time. Yeah, and this standard disclaimer, anything I mentioned, active ingredient, commercial name, trade name, are for research purpose only and for information only. Does not, endorse anything that they are only products that we recommend folks to use, right? So just kind of just take a 10 seconds to read this. Just I have to put it there just for UH standard disclaimer. Acknowledgement of the funding agencies, like I said, past and former and current lab members who contributed to this research, and also Cornell Hills for hosting us for 2 years, and any other folks who participate or helped in one way or another.